Hello, Biology 230. This is Dr. Chastity Bradford, an assistant professor in the Department of Biology at Tuskegee University in Tuskegee, Alabama. And we are covering Chapter 6.4, which is Concept 6.4, and this is the lecture 6.4b. So we're continuing to tour the cell. And now we're going into functions of the ER and how the ER distributes transport vesicles. So what are vesicles? Vesicles are small membrane-bound sacs, you see here. The contents within the vesicles are transported to another membrane surface. And also, these vesicles can fuse with membranes and deliver their content. So what you're seeing here is vesicles being formed, and then they're budding off, and then traveling to their destination. This is one vesicle here that's in transit to the Golgi. What is the Golgi? The Golgi apparatus is well known as the post office of a cell. It's the shipping and the receiving center. It consists of a number of flattened membranous sacs called cisternae. The Golgi functions to modify products of the endoplasmic reticulum. It functions to manufacture certain macromolecules, and this term should be very familiar to you now. It functions to sort and package materials into transport vesicles. And there are two faces of the Golgi. There's a cis face, and it's closer to the endoplasmic reticulum. And this is the receiving end of the Golgi. And there's also a trans face, and it's on the side closest to the membrane. And it is the shipping side of the Golgi. And so what's depicted here is the cis face. So that means that the ER would be on this side to the left. And then the trans face of the Golgi. So you see a transport vesicle coming from the endoplasmic reticulum. And it comes to the cis face of the Golgi. And in the Golgi, it may be modified. Okay? It's sorted and packaged, if you will. And then once it's ready, these proteins that are now inside these transport vesicles will then go to their destination. Their destination could be another plasma membrane. Their destination could actually be to come back to the Golgi and become part of the Golgi's membrane. Okay? So let's look at this animation. And in this animation, you'll notice various steps. Okay? And I want you to really look at how the ER and the Golgi work together. So as I mentioned, this is the Golgi, and what you see highlighted in orange is the endoplasmic reticulum. And now we're highlighting or magnifying the ER and the Golgi. And so you see these vesicles that are budding off from the ER, and they're traveling to the Golgi. Okay? And if you recall, this then is the cis face of the Golgi, and this then would be the trans face of the Golgi. Okay? So both the Golgi and the ER are components of the endomembrane system. And the primary function of these organelles is to produce mature proteins, which are inside of these vesicles. And these mature proteins then will be exported from the cell, and they can be incorporated into the membrane itself of these new cells, or they can be moved into other parts of the endomembrane system. As I mentioned, they could actually come back and become part of this membrane of the Golgi. Okay. So now let's go to a 2D perspective of the ER and the Golgi. So now, this is the ER, and new proteins that are made here in the ER, and this is a protein, for example. We talked about this, that these proteins, they become embedded in the membrane, and in the ER, there are enzymes that are here. And what these enzymes will do is they will add sugar groups. So once again, these proteins are being modified in the ER. They're adding sugar groups to some of the proteins. And guess what they become? Glycoproteins. Let's continue. So now, let's take a look at what happens when that vesicle is transported to the Golgi. Did you see it? And then it fused with the Golgi. See if we can go back. Allow you to see that one more time. So you see this vesicle fusing with the Golgi. And of course, it's bringing with it these modified proteins. So these vesicles, they're fusing with the Golgi, and now it's adding to the volume of the Golgi, and of course, adding protein content. Okay. There are cisternae, so these regions are called cisternae. Of course, this is the cis face, and this is the trans face of the Golgi, and the middle region, or the medial region of the Golgi. Okay? So remember that the part closest to the ER is the cis face. And that sounds like someone is going to the ER, but not the endoplasmic reticulum, but the emergency room. So remember, ER stands for endoplasmic reticulum. Okay? And then this is the trans region, and the trans region is the region nearest the plasma membrane. Okay? Let's continue. So what you're seeing now is the Golgi being highlighted. And notice that the proteins that are here, they bud off, and then they fused into that medial region of the Golgi. Okay? Let's go back and review that again. So notice how we're highlighting the Golgi, and now these proteins, they're budding off, being transported, and they now become part of the medial region of the Golgi. So their further processing is taking place. Okay? And each compartment, so these are cisternae, so each compartment of the Golgi contains enzymes. And one of the functions of the Golgi, you remember, is to modify, synthesize, and to store proteins. And so these proteins are modified, okay, by enzymes. And in this particular case, let's continue here, a sugar residue is cleaved. Okay. So now, notice how it became part of this other region. Now it continues to be transported. Okay. And now it buds off and it leaves the Golgi and traveling to its final destination. And in this case, these proteins that were made and modified, they were targeted to the plasma membrane to be secreted outside of the cell. Okay? So it's important that you understand that the Golgi works in concert. So this endomembrane system, they all work in concert. And the Golgi is a network of membranes in the cytoplasm whose function is to modify, synthesize, sort, and store proteins. Okay. So now let's continue. So for a review, if you recall, the nuclear envelope is continuous with the ER. This is the rough ER because you notice it has the ribosomes attached. And then over here you have smooth endoplasmic reticulum. And what you see here is that there is a vesicle here that buds off from the rough ER, from the smooth ER. and it contains proteins. Remember, those proteins were modified there in the rough ER. They're transported to the cis face of the Golgi. Now, all of this is taking place in the cytoplasm. And then they're modified here. They're synthesized. They're sorted. Okay? And then when they get to the trans face, once they're sorted and we know they're tagged and the body knows the destination for these proteins, they bud off from this trans face. And depending on the type of protein, if it's a glycoprotein, it's modified in a way that sugar monomers are removed to produce carbs. Okay? And then they're then secreted outside the cell. Okay? So notice how this secretory vesicle, or this transport vesicle, secretory in that it is, its function is to secrete. Okay? Its function is to release proteins that will be secreted, so therefore it's a secretory vesicle. It will fuse with this plasma membrane here. And then these proteins that were inside will be expelled or secreted. Okay? Lysosomes are also components of this system. And the lysosomes are, some people call them the garbage truck of the cell. Others may call them both garbage and recycling trucks of the cell. And I'll explain to you why. They're the sites of cellular digestion. And what you have here in this image is the image of a lysosome. And you see that the material is being digested. Okay? Now, how does this all take place? It's a lot of words here. It's a lot of things for you to learn and to study. So lysosomes are sacs of hydrolytic enzymes, and they're responsible for degrading the old, if you will. They retire the old organelles. They're responsible for degrading food items. And inside of these lysosomes is an acidic microenvironment, a pH of about 5. Okay? And you recall anything less than 7 is acidic. Okay? And so the environment prefers to be at a pH of 5. Now, so inside of the lysosomes, the lysosomal enzymes, they're responsible for hydrolyzing proteins, fats, polysaccharides, and nucleic acids. Okay? Now, they contain these digestive enzymes that will break down the cellular waste and debris, and also nutrients. They'll break down some nutrients that maybe the cell needs to use. So these are different examples of how this takes place. So if this is our cell, and I'll show you an animation in a minute, this is a particle. Let's say this is a particle here. This is a damaged organelle, and here's a lysosome. So the lysosome and that damaged organelle will come together, and the enzymes within the lysosome will degrade or lyse the damaged organelle. And that's how we remove these damaged or spent organelles. The same thing can happen for food particles that need to be digested. The enzymes within the lysosomes will bind to those particles, and then those particles will be digested. Now, there's another process that you should become very familiar with, and it's called receptor-mediated endocytosis. Now, this takes place with certain macromolecules, okay? And what you see here is, this is an endosome, and it's fusing with a vesicle from the Golgi. Okay? Now we have a late endosome, and that late endosome then becomes a lysosome. Okay? So receptor-mediated endocytosis. Let's now look at the lysosome animation. So lysosomes, they're membrane-bound vesicles. So here's the lysosome. They contain these hydrolytic enzymes. Remember, they like a pH of about 5. And these enzymes will digest particles or cells that are taken into the cell by phagocytosis. And these hydrolytic enzymes that degrade the proteins, nucleic acid, lipids, and carbs, they're formed in the ER and then transported, see them, formed in the ER and then transported to the Golgi. See how these lysosomes then arise from the Golgi, and then see, now you have particles like viruses or bacteria ingested by phagocytosis. Now this lysosome will fuse with this bacteria or the virus and degrade it. This lysosome can fuse with an organelle that may be defective or worn out or old, and it destroys it. Okay? And this is how our body maintains and regulates our system. So let's look at this again. So here we have lysosomes. They contain these hydrolytic enzymes. Okay, remember the pH of 5. And these lysosomes are responsible for digesting particles or cells taken into the cell by phagocytosis. See how these enzymes are produced here in the ER. Because remember, enzymes are proteins as well. They bud off. They come to the cis face of the Golgi. They fuse with the Golgi apparatus. And then once they're modified and they're ready, these lysosomes then come from the trans side, bud off of the trans side. This is some bacteria. And these lysosomes fuse with this bacteria. So now we have what's called a phagosome, degrading it, protecting our system. Lysosomes can bind with these dead organelles, old defective organelles, worn out mitochondria, destroying them and protecting our system. It is amazing. Amazing how we are designed. So another part of this system, this endomembrane system, is a vacuole. Now, there are different types of vacuoles, but let's define them. They're membrane-bound storage areas for resources. They have no internal structure, and they function to store undigested nutrients, and they're not vesicles, vacuoles. And how are they different? They differ by their size, so they're much bigger, and their function. Remember the lysosomes, they had these enzymes, and they're the digestive system. They're more for digestion and degrading old organelles or worn out organelles, or getting rid of bacteria or some type of virus that tries to invade our system. But vacuoles, on the other hand, they're more for storage. Okay, and so what you see here, depicted down here, is a lysosome. Okay, remember they have these digestive enzymes, and you see a food vacuole. So if this needs to be digested, it can fuse with the lysosome, and then digestion will occur. This is a micrograph of a vacuole. Okay, so see how huge these vacuoles are and how empty they are. So they have no internal structure. Okay, we just reviewed the food vacuole is one type of vacuole. There are contractile vacuoles, and these exist within some animal cells and protists, and they're responsible for removing excess water. And in chapters to follow, you'll understand how important and vital osmosis is and the use of these vacuoles are in osmosis. Another type of vacuole that's found in plant cells, primarily in plants, are central vacuoles. And they are 90% of the cell volume, and they're mostly used for plant storage. So you would really be able to recognize a central vacuole. You should be able to recognize a central vacuole image of one, because look how much space they take up, okay, in a plant cell. There are diverse maintenance compartments, okay, and remember, compared to the vesicles, they're bigger. A plant cell or a fungal cell may have one or they may have many vacuoles. And we just reviewed food vacuoles and how they're formed. They're formed by phagocytosis. Review contractile vacuoles that are found in protists, used for pumping out excess water, and central vacuoles, found in many mature plant cells. They're formed for storage, used for storage. They function to hold organic compounds and water. So by now you should be able to summarize the path of proteins through membrane. So if given an example of a type of protein that you know would be secreted, in a scenario, you should be able to demonstrate to us that you understand the path paths that these proteins would take that are destined to become secreted. Once they are modified in the ER, they bud off, transport vesicles are formed, they bud off, and they become fused to the Golgi. They come to the cis side of the Golgi apparatus. Okay? They can become modified here within the Golgi. Okay? As you notice here, there's a vesicle that contains proteins that are leaving the Golgi, and these proteins, this vesicle fuses with the cell membrane, and then these proteins are secreted. So this would be a secretory vesicle, and these proteins are secreted. But also, the Golgi produces lysosomes. So this is an example of a lysosome budding off from the Golgi. Okay? And then vacuoles are also produced in the Golgi. Okay? And this is an example of a vacuole. And recall, there are different types of vacuoles. The rough ER, it synthesizes membrane-bound proteins, and it creates transport vesicles that fuse with the Golgi, producing membranes for the cell, abundant in cells that secrete protein. So if there is a cell that you know is responsible for secreting proteins, then you'll have a lot of rough ER there. Okay? Making phospholipids, that's something else. Synthesizing phospholipids is another role of the rough ER. What about the Golgi? You recall, the Golgi is a network of membranes within the cytoplasm, and it's responsible for modifying, synthesizing, sorting proteins, and storing them. Okay? As these proteins move throughout this Golgi apparatus, they're being modified. And this modification determines their destination, whether or not they become lysosomes, whether or not these proteins become vacuoles, whether or not these proteins are destined to become secreted. All of this takes place in the Golgi apparatus. These vesicles then are sent to their final destination.